So good morning. My name's Tony Blau. I'm one of the hematologists here. Bill Bremner asked me to introduce today's speaker at Medical Grand Rounds because he and a bunch of residents, including our chief residents, are at the meeting in Carmel. I told him it'd be a real pleasure to introduce Stephen because I know Stephen Friend really, I think his signature mark is as a visionary. Stephen has a very clear vision for the future, and he's been able to demonstrate not only the vision and the promise, but also the ability to deliver on that promise. So he's a pediatric hematologist oncologist. In his research lab experience at Harvard, he described one of the first oncogenes, came to Seattle in the 90s and started a commercial company called Rosetta Informatics that was sold to Merck and then took a very senior leadership position heading oncology research at Merck. And now for the last several years is president of a not-for-profit organization that many of you will not have heard of until today called Sage Bionetworks. And so Stephen's, I think, gift is for understanding what needs to be done to move medicine to the next level. And the topic of his discussion today, I think, is really going to allow people who probably don't think about this much to really begin to appreciate what the future of medicine is going to look like, and that future is not all that distant. And I think one of the main messages that I really appreciate that Stephen is a main advocate for is that the obstacles to preventing us to realizing the potential of technology and computation, those obstacles are not technical obstacles. And one of his main themes is the need to find ways for people to share. And so it's an immense pleasure to have Stephen. Thanks very much for coming.
Nice to be back. At some point in the 90s, I was actually in the pathology department, Larry Loeb's pathology department here at the UW. So it's fun to be back in that sense. Most people don't know that. The other thing to throw out before I begin is as I'm giving this description today, put in the back of your head, sort of like running along simultaneously, what happened in physics during the 20th century in particular. What happened is people began to have insights that led to what could be done with nuclear fission, and the shift from the laboratory physicist to team-based physicist to CERN. So keep that running in the back of your head when you're doing a reality check with what I'm going to do today. I've got 5 parts. I have no slide numbers, but you can tell where I am because I'm going to go through a description I think is important to start with, which has to do with looking at complexity, where we really are in that. Then I'm going to shift over to looking at what I think are a pretty awesome portfolio, belt of tools that have happened in the last 10 years. Then how we're engaging some of those through to a project that isn't yet launched, but I've been thinking a fair amount about as a way to end. So let's go to scale of complexity. And to give you a sense on scale of complexity, I'm going to give you 4 examples of where many, certainly including myself, were naive about what was going on, what you should anticipate as we're starting to get information. So let's go to DNA alterations. And if you dial back 30 years, that was the beginning of an era which, as I think some of you will remember, was where there was this sort of sensational front of New York Times. I've found this disease is caused by this DNA alteration, this disease, whether it was CF, whether it was breast cancer. It was a time when scientists as optimists were giving a description of the fact that we were uncovering the component list that should then be able to be used to be able to impact patients. This is Frank McCormick. If he was a better race car driver, he's a pretty good race car driver actually, but if he was a better race car driver, he wouldn't be the head of the cancer center in San Francisco. There's a nice article that he put out this last year that I want you to compare contrast to what I just said. So this diagram, sitting over here in the black, has little triangles, and they're components of a signaling pathway. It's the RAS signaling pathway. And the paper that he put out should have shocked people. It did me, which is when you modified components of that pathway that sits, in sort of like some absurd, simplistic diagram up there, what happens is that the function of every other component on that diagram changes its function. So my point here is that we, whether it's as physicians or as scientists, would like to have some pull-down chart that says this, goes to that, and we've learned these, and we're going to make a drug against that. But what this chart tells you is when you have alterations, imagine there are lots of them in cancer. When you have alterations, those alterations change the function of other components. It's not just I'm going to draw a diagram of a radio and I pulled out a transistor and it functions differently. It changes the function of other components. So that's an example of how naive we have been. Second example, easy to again present, is to look at expression profiles. When we were up in Kirkland as Rosetta, working with a number of labs, Ron Davis, others, at that time in yeast, it was possible to go and make knockouts of function across the genome and do it for the entire genome in yeast. And at the same time, you could go and make a little snapshot, like frames in a movie, a snapshot of what happens when I knock this out, when I knock that out. And it became apparent that that's pretty cool. That what you could do then was to build a compendium of knowns and use that compendium of knowns to query unknowns. And that's in fact what this paper showed. We could stick in FK506 or cyclosporin, drugs that were working in people, and we could see how related they were and what they did simply by going into this compendium, using patterns of what went on without knowing anything about why. A different way of doing science. And that made it seem as if, oh, it's going to be exciting, right? We'll build a compendium for human biology. We'll be able to figure out drugs with new purposes, et cetera, things like that. I think you know the reality. Go to any disease you care about. Go to Alzheimer's, go to cancer, go to diabetes. And what you will see is, for any one disease, a series of papers that say, here is my favorite classification scheme, using that concept of a compendium, building up a set of knowns, and saying breast cancer can be divided into 4 categories. Or if I go to Crohn's disease, I can look and see categories. And so something shattered here again, right? The same way I talked about it shattering for the DNA, it shattered here with regard to the fact that when you looked inside the cell, when you could hang thousands of microphones and listen to what was going on, you got different answers. Not everyone getting the same answer, not because they were sloppy, not because they were criminal, but because in fact the context was not the same when we thought that it was. So I'm building a story on context. The third is some people could go, oh, you guys, I left out the protein component, which works exactly the same way. Some people go, oh, you guys are simple. If you just look at function, that would clean things up. If you could look at function, then maybe you'd see those patterns. Maybe you could see something that was coherent. And in this paper, there really is an outgrowth of beautiful time spent with Lee Hartwell and others here in Seattle. This one was with Alan Olaf. And we went and showed the power of using synthetic lethal strategies to take what was in the tumor and go and find a drug that would selectively kill tumor cells over normal cells, just in the way that you do in yeast. And again, we thought, we've risen to a level where we're going to be able to operate. We've gotten to a level where we can be able to make insights that will have an impact. That should allow us to make drugs which are synthetic lethal, and then put it into those individuals that have that defect, and watch them work. This is going to tie into the fifth part of the talk. So what happens here? Here are four good friends of mine. They took a nice example of this. They took the RAS synthetic lethal screen. A lot of patients in cancer have RAS alterations. They thought, we're going to make the powerful drugs. We're going to run a synthetic lethal RAS screen. We're going to come up with genes that are synthetic lethal. We'll use those as targets. We'll make a big impact. Each person here, whether it was Todd Gallup or others here like Gary Gilliland, got a different answer. What's going on here? Every single one of these papers has a different answer of what's synthetic lethal. There's a little bit of an overlap. So I gave you a DNA picture, I gave you an RNA picture, I gave you a synthetic lethal picture, and I could do the same for drug trials. Here is a set of 5 targets, which inside of industry we built beautiful inhibitors, nanomolar inhibitors, very selective, and expected those to go into clinic with all the knowledge that we had, some of these driven by synthetic lethal concepts. 2 of them worked. 2 of them worked like we expected. Okay, hundreds of millions of dollars down the drain. This is why it costs billions of dollars to develop drugs. So this is because when we go, and whether it's teaching our students or thinking about how we're going to use drugs in the clinic, here's EGFR, there's HER2. We have this way of picturing that that is the way the cell is working in this sort of stick figure, ball and stick way of thinking. And I like the way Eric Schott put it. He said, our brains are hardwired for the narrative. We love storytelling. We like to go this, goes to this, goes to that. And guess what? Our cells purposely don't work that way, okay? I can't show those 3 dimensions that are there, but most of the control circuitry, most of the things you might be interested in modifying are not sitting there right along the pathway, that instead for any diagram that we draw, the interesting thing is how related everything is to everything else. So why have I told you that? Why have I gone through that whole description of complexity? It's because I feel that we have to be honest with ourselves and realize that we are no better than the 18th century chemists who were truly only alchemists, who had big files, they had big sets of relationships. I'm sure they had the equivalent of all these large papers that we have in chemistry. They were missing the periodic table. So one of the things I want you to think about is, what does it take to get to the rules that govern what go on between components in the cell? Not, I found this and this is associated with that. But what is it going to take to figure out the rules? Because until we understand some of those rules, we're just going to be dabbling and filling up giant databases. And it's particularly awkward because much of our scientists, we say, you should do hypothesis-driven research, right? You should have your hypothesis, grid down all the things that you can, have a very few set of unknowns, and do your experiment. And that's correct, okay? You can do that the same way the physicists in the 1920s did that type of experiment on their lab bench. The problem is that there is no one right answer. The problem is that often, in order to get into a one-name journal, you have to take something that's a particular and go, and therefore, and in the therefore, I now know this about diabetes, or I know this about cancer. The reason why that's a crime is that in doing that generalization, often things that are to the left and right of it that are completely relevant are seen as irrelevant. Problem is, we get to thinking, now we know this, now we know that, now we know if we give this patient this drug, it will work in this way. So that intro is because I think we have to have eyes open, we have to embrace the complexity, and I want to give you 6 tools that I think are capable together of tackling that level of complexity that takes us beyond the storytelling, the narratives. But first, you know well, the ability to generate data. Anyone's hyperbole from a couple of years ago has sort of been erased by time. I'm not going to even talk about the way that data can be generated. I think you're probably well aware of that. You might be aware of the power that has come in the last 10 years by a series of probably 100 papers, I don't think more, that have been done by a dozen or so labs. So this is more immature, but where people realized that you could build networks that actually were not classical pathways, but you could build relationships, priors, and use tools that large data science efforts, whether they're in business or whether they're in physics, have used, because we have enough data now in biology to be able to apply these network modeling approaches. I'm going to show you one a little later in the lecture. But remember, this changed it, okay? Got us off of the two-dimensional ball and stick diagrams, where you had to have diagrams like that. The third, I think you're all aware of, but it's very important, and that is that don't forget that in the 80s, early 90s, if you had data, that doesn't mean that you could share it with another. The ability to actually have the internet, to put things up in the cloud, to be able to say, I want to look at your data, you can look at mine, was running hard disks around. And for the stuff I'm going to go into, it's actually really important that we have that capability around us. The next, which is a little reference back to the physicist that I was talking about at the beginning, is that some funding groups are beginning to wake up to the fact that you should not say to a scientist, do you have a great idea? Can you generate your own data? Can you do your own analysis? And then a year or two later publish. And that's the way you're going to make your career. I think really importantly, we're beginning to get beyond these closed loop strategies and recognize that the person that generates the data is probably not the best person. Okay, this was hard. But people are beginning to realize that the person to analyze the data is often the best person is not the person who generated it. That it takes different skills, it takes different knowledge, and that you don't have to generate data to be able to work on a question. That uncoupling of that loop is in a paper that just came out last year that we wrote in Nature Biotechnology, referring to Metcalfe's law. The next is that citizens, patients, in the last 3 to 5 years have begun to going, hey, can't I be a part of this team? Can't I collect my own data and give it to you? How is it that instead of having someone who has a neurodegenerative disease, twice a year they go to a doctor that says this is your new level of your disease? What happens if you allowed citizens to be collecting objective sensor data, subjective data? And I'm going to go into that near the end. But this concept that actually there are very inefficient ways to enroll tens of thousands of patients, not paying every patient $1,000 because they're going into a trial, okay, comes up and sort of shears the usual way that we're looking at collecting information. This is a beautiful site of Crohn's disease. Notice not chronology, but chronology, in which they're going out and looking at what drugs work in each other and what they should do, what they shouldn't do. This is run by Sean Arenz. And then homegrown here at the UW. The next thing is, can you get citizens who have no postdoc, have no graduate degree, can you get them to actually carry out tasks that usually you go to get a graduate degree or a postdoc? Can you make it so you don't have to go to graduate school and get a postdoc and be an expert, right? What is it that you could do that separates that? I'm going to sequester you in a little tower for a while, and then you're going to come out anointed. And I think Zoran, you know the work of David Baker and Foldit, Adrian Toole. There are many examples now. The winner, for example, in case you don't know this, for one of these challenges on RNA folding was a housewife in Surrey who she would do her work by day, put her kids to bed, and then she'd spend 3 to 4 hours working on rules for electrostatic interactions and go to bed. And she beat all the graduate students. She beat all the other people in the world. Okay, so there's no reason. Okay, no reason. If you have passion, okay, and you work as an effort, there's no reason you have to go and take a course in something and get anointed and say, you now know how to do that. So the 6 ways that we've been thinking about is all this data that's beginning to flow on omics, the network modeling, the IT infrastructure that allows you to put it so you can get to it, the uncoupling of the linkage of those that generate from those that analyze, the nascent movement for patients to now start being part of that, generating and analyzing the data, and then this concept that citizens can actually be an expert without necessarily going through an academic and going into debt. So 4.5 years ago, almost 5 years ago, I peeled off from the worlds that I had lived in and said, let's form a group. We didn't form a group because someone bestowed a lot of money on us. We just said, we'll be a group. This is a nonprofit organization. Sits inside of the Fred Hutch in the Arnold building. And there are about 45 of us. And what we're interested in doing is recognizing that we think biomedical research is about to fundamentally change. What can we do to run little pilots, to run little test projects, to test how open, collaborative can you get teams to work, and how do you get beyond the current guilds of experts in order to get discoveries out faster? So sagebase.org, you can get onto the site. You can look at the projects that we're doing. For those that are in the audience thinking I was going to talk mostly about doing a scientific experiment, I want to give one quick example. This work done by Chris Gaitari, who is with us now at the Allen Brain Institute, was lead author on this paper that was in Cell last year. And I like this story because it was so simple. So we didn't generate any of the data. Okay, we got someone to do the validation. This project took about 1 year, and I think it fundamentally changed the way we look at Alzheimer's disease. So all we did here was we took a bunch of genes and their expression, and we basically aggregated them into modules that were just co-expression modules. I've color-coded some of them, but the point is all that was done here was say, if this changes are similar to those changes or similar to those changes, simply looking at similarity of RNA expression. Then what we did was we said, let's prioritize and say some of these are more important for this disease or others because in a particular disease, Alzheimer's, the genes that were in those modules were in fact had previously been shown to be more involved in this or that component of the shift from normal to clinical Alzheimer's disease. So in this instance, it was basically using the clinical information in terms of which patient had what severity of disease that we then layered in on that. And then we went to genetic information, and we basically incorporated eSNP information to actually look at how those modules might be related to each other. And what you get is a system that had nothing to do with what we expected. So at Merck, I also had the neurology discovery group reporting in for a while, and we studied A-beta, we studied tau, we studied tangles, we studied this, we studied that. Okay, this diagram here should look familiar to people in immunology. It's the complement, the FC receptor, MHC, chemokines, the units that were actually driving Alzheimer's. The components that were the nodes that we could see actually were all about an inflammatory process. Here's neurodegeneration. This is about an inflammatory process. So we linked up with a group in Germany that could look at microglia. It could do experiments with or without, and basically started knocking out the components that were at the nodes. And one of the nodes that stood out is this TYRO-BP that's part of the TREM2 pathway down to PI3 kinase. And you may know this story. This is one of 3 papers last year that shifted the interest in the field in Alzheimer's to the roles of the inflammatory system and how does that intersect with what was going on with ApoE4, et cetera, et cetera. For work that we do in oncology, Adam Margolin, who has been a part of our effort at SAGE and is now headed to Oregon Health Sciences, built up a group that had started from work that he was doing at the Broad that started saying, how in cancer could we do this type of analysis? And for those that are interested, I look at the audience, and I think there may be 6 to 8 people in the audience that are interested in this. I'd be glad to go through this later. But there are ways to figure out what are the best machine learning approaches, how to go and build predictive models, And one paper that you can go look up was one that was on the Cancer Cell Line Encyclopedia at the Broad, where the group led by Adam went in and said, can I find the driver genes for the sensitivity to a set of a dozen drugs? So he said, 12 drugs, I've got the encyclopedia. And this beautiful paper went and showed that 11 of those 12 particular targets, you could find a driver gene and then validate that driver gene that was responsible for cell line sensitivity. That's turned into a fairly large effort over at Sage, looking at predicting cellular phenotypes to guide therapeutic strategies. And I just want to leave you with one sort of beautiful idea that they have working on. This is the work of Mehmet Gonen. And what he realized, which is well known again to people who work in financial markets, is that if you have a very big database of information, you can train on that very large database and then have a transfer of that learning by using Bayesian kernel learning technologies that allow you to learn on a semi-relevant data set and still come in and move that into the one that you're interested in. And that's actually how Mehmet won one of the challenges that was one of our worldwide challenges. But just to give you a specific example, we're getting ready to submit this for publication. What Mehmet did was he took the TCGA data, where there's a lot of information about expression, but also about mutations, and used it to impute in the cell line data in CCLE what was going on there to increase the predictive power of looking at driver genes in there. And then what I really like is he went and basically imputed the underlying mutations that were causing the sensitivity by using the large database, TCGA, coming over. So he imputed the mutations that were sitting in the smaller database from the large database. And why is that important? If you're going to be treating a patient, you're not going to have a lot of information about what's going on in that one patient. But imagine you could develop a way where you could use transfer learning tools to actually take a lot of data that was known in other dimensions and reflect that over. So the thing that we found was keeping research scientists from working together is that they didn't want to talk about their work until they knew they had their first author paper that could get them tenure or get them whatever reward on the academic ladder. And the reason why that's hard is that the interval of time between when someone knows something and when they get credit for it is basically measured not in days, but in years. And so we have been communicating, the currency of communication in science has been journal articles. And yet at the same time, in the last 10 years, software engineers and other groups, in some ways the physicists and mathematicians used archive, but the thing that we were jealous of and decided to emulate was this approach that software engineers have taken called GitHub. And what GitHub does is it allows one to track every code change, every issue. It allows you to fork what's going on so that 100 people that have never met each other can work on a project. You can make it so your small group is working on it, but the point is the credit. You can track who did what in that system. And this system, which started out about 4 or 5 years ago, not too much before when we started Sage, now is so prevalent that millions of projects, millions of people are using it. And now when you go and apply, let's say you go apply at Microsoft for a job or Amazon, they don't ask for a CV, okay? What they want to see is what you did, not what you say you did. So what we're interested in doing in biomedical research is getting it to you getting credit for what you actually did. Not the head of the lab, not because you were on the paper listed, but what did you really do? Okay, and we think that that's how you should get sort of the recognition. And so we built out a version of GitHub called Synapse, which essentially can sit on. So all the code for Synapse is open, and it sits on GitHub. But Synapse is a place where scientists, biologists, computational biologists can share what's going on, analyze who's doing what, and very importantly, have So it can run sort of private and public so projects or public projects. It is a place where you don't have to use any particular tool. You can use Google or Python or R. You can run it on any platform. But where I was headed was, what it gives you is provenance. You can have the transparency and reproducibility that we think is missing. When you submit or you get to review a paper that someone does in computational biology, you can't figure out what the person did, okay? No supplementary material is capable of actually giving you an insight. And so we want to make it so that there's transparency, reproducibility, and credit that you can give to track who did what all the way up and down the ladder. So I want to give you 2 or 3 examples of how we're currently using this. The first is there's a very large project called TCGA, which has 12 tumor types and 6 modeling profiles, so molecular profilings from DNA and RNA, et cetera. And what we did, similar to like the ENCODE project, was we took 30 institutions, 150 researchers, and said, why don't you share all your data in real time, the way the genomic scientists for some time have learned to share their time, but now why don't you share your clinical, your other data in real time, and say what you're doing, get credit for that. And at the beginning of this interval, we said, give the paper you're trying to write. Give us a description of what it is that you're trying, and people will join your team to help you do it, or you may recruit people from there. But the whole point was they worked as sort of within a walled garden as a community. And the fun thing about this project is that it took 9 months to go from forming teams to getting accepted papers. So that's about the time that usually you're going between when you're writing the paper and when you're getting submitted. So that was a significant acceleration. You can go and look in Nature Genetics at the article that describes how this work was done. Larson Omberg did that. Justin Ganay is doing another project that I think gives you an example of how biomedical research can be done. There were 7 groups who were working on colon cancer, building classifiers, saying, I want to build a classifier the way we have Oncotype DX and MammaPrint for breast cancer. Let's build classifiers. And the problem, back to what I said at the beginning, is the first 3 groups got slightly overlapping answers. They were not the same. Right? So one classifier said, let's make 3 categories. One said 5, one said 8. And first group sort of got a pretty good journal article. The second one, hey, one's already been published. The third, by the time that was over, there were 4 or 5 other beautiful sets of clinical data with genetics and people who could do algorithms, and no one was interested. They said, we've already seen this. So we collected all those 7 groups, and Justin started, in fact, I think today, starting at 7 a.m., he had one of his Thursday calls, every week to every other week, sitting down and put all of the data from all the patients in 7 different countries. So it was all on Synapse, for the clinical, the genetic information, and said, let's work together to build a classifier that will be able to be used by many. So what I see this is a method by which to accelerate the adoption of interoperability standards. And if clinical groups could do that, then you could go, oh, I want to put my data in, I want to increase the power to do that. And in the most recent example of what we're doing, and this is just something that we started in the last month and a half, we went and looked at someone who I think is likely to be given many of the prizes that get given in medicine for this paper that came out a couple of weeks ago. So Ali Kalimani is working on AML. He works in Helsinki. And what he did was something you can't do in breast cancer, you can't do in colon cancer. But in AML, you can look at the whole tumor all the time. You can take a sample, you can get it out, you can look at it as often as you want. And more importantly, you can grow it at the same time as the patient is there. So what he realized, and the group did in a really awesome way, was that you could do these ex vivo analyses and get the sensitivity to drugs at the same time he was building his patterns, and built a loop where the patient who was getting was refractory to other therapies could go in, do that analysis, and in real time showed how he could get that information back. So I know that Charles Sawyers, I know that Jose Baselga, I know that people at the UW I know people all over the world who are doing this. This guy trumped everyone in what he did. So because we know his group, Jerry Radich is here, Soleil is here, Brian Drucker is Oregon, and Phoenix, is we said, let's form a federation, very much like a Star Trek federation of labs that have said there's a big enemy out there, and let's work together and let's share all of our data, let's share all of our patient information, let's run the informatics, and let's run the equivalent of virtual tumor boards where when someone comes up who's about to be treated, all 4 of those centers are sharing their same bioinformatics tools, they're sharing the information that has to do with making a decision. And I'm happy to say that these groups all these people are far enough in their careers that they're not trying to get tenure, right? And so it takes finding people who are ready to sort of suck it up, and the people in their labs, I think, realizing the benefit to patients, but also the papers that will come out. So now to the next to last part, which is that's working within scientists who are assigned, right? I did the examples of colon cancer, TCGA, and those are assigned walled garden groups. What happens if you could get data and throw it up in the cloud and let anyone in the world work on it and not care what degree they had or where they came from, and just have an analysis? Can you get a better ROC curve? Can you do better than another? And so last year, I guess it's now like a year and a half ago, we were lucky enough to get these massive data sets and articles that had just come out in Nature of thousands of women with breast cancer in Canada and the UK from Carlos and Sam. Google gave us free compute space here in town, Joe Hellerstein did, and allowed us to put that up. And then we went to Science Translation Medicine and said, let's blow up peer review. Instead of saying we're going to figure out whether this person likes that, why don't we say the person that should be getting the first author paper is the one that beats everyone else in the world, right? Isn't that peer review? And so we worked with them to make it so that the people who were entering this knew they had the ability to have a first name author in an important high impact journal, working with data they didn't generate, but just and didn't care where they had trained. And as you may know if you've heard this story, the group that won it was a set of electrical engineers and physicists that did not know cancer biology. They used approaches that we wouldn't have thought of. They worked closely with clinicians, but they didn't have that knowledge. There's a nice article that's on the challenge, their article. There was a cover story about that. And we realized that we needed to fuse the dream challenges that were being run at IBM for the last 6 years with SAGE. So we teamed up, and I think Gustavo Stolovitsky, who's part of this, who I work with, is actually giving a talk maybe tomorrow today or tomorrow at the UW. He's in town to work here and there. I want to give you 2 quick examples of challenges that we've now gone to, which is we said, why don't we take patients who are getting approved drugs and see if we can now figure out who shouldn't be who shouldn't be given those drugs. Okay. So we thought a good example would be to go and take the anti-TNF therapies for RA, because only about 40% of the patients respond. And we thought, let's see if we can find an algorithm, put it out to anyone in the world, and see whether you can identify the extreme non-responder that really shouldn't, less than a 15% chance of responding. We got 4 companies to work with us because they want new drugs to come in, so they were willing to do that. We have 10 clinical trials, and a description of this is sitting in Nature Genetics. It's about to launch. We have 100+ teams that are poised, waiting to enter the data, and we haven't even started the thing yet. I bring this up because this time we shifted the challenge prize. There still is one about publication, but we said, let's give the person who wins that challenge their classifier, let's run it in as a validation into a clinical trial. And let's have the clinical trial be data that comes directly from patients. So we went to the FDA, we went to the EMA and said, can you help us build up a classifier where the data is not coming from an industry trial, not coming from a university trial, but coming directly up from patients with the help of the nonprofit Status and the Arthritis Foundation. We're getting ready to run a validation trial that will follow this, but it's completely being done outside of the usual complex. I don't have time to go through a nice challenge that just opened up about mutation calling, because I wanted to highlight this challenge that's coming together. Andrew Trister, that works with us, that is in radiation oncology here at the UW, and Zoran, you may know, are in ways part of this. But I want you to think about the following challenge. You probably know that when someone is diagnosed with melanoma, I'm not talking about they get their biopsy, I'm talking about when the pathologist looks at the slide. We assume for all diseases that the pathologist, I can say this because I was in the pathology department here, is the God that sets the gold standard. The pathologist says, this is what's going on, right? And then we tie all of our molecular analyses to that. What happens if the pathologist is actually unable to give really rigorous answers and has to actually sort of hem and haw a little bit? And in fact, when you say the diagnosis for melanoma, some people think 9 out of 10 patients who get given that diagnosis would never have gone on to die of that disease. But let's make it 4 out of 5. So the question is, if you don't have someone's head and going, I've looked at this slide, I'm going to call it, right? But imagine that you have a knowledge expert, which is driven off of rigorous ways of looking at patterns, was actually backing up that person. Maybe you could get a more gold standard level of information for any disease. And so we decided to work on melanoma because we've now collected over and this is the work of Andrew, over 1,500 cases that are up in the cloud, and they have a yes or no died of the disease. So we're able to look out X years, put that tag of outcome on it. We want to build that to 10,000. We want to build it beyond 10,000. What's really nice is DARPA, the people who built GPS and built the internet, are very interested in using their pattern recognition tools that have nothing to do with medicine to see what they can find and how they would do the interrogation of the slides. We've been talking to Zoran. I've got a conversation today or tomorrow to see whether one of his teams would come in. We're going to do a dream challenge on it. There's a group of medical informaticists that are image sort of challenge runners that are going to work on that. So then I'm going to turn now to the fact that we still feel as if we have a problem, and that is that citizens who really want to get engaged in this are just as voyeurs, just at the outside. So whether you're really famous or not well known, or you have a kid that has a particular disorder, it would be nice to have those people engaged. And it also will really help us because we're getting all this genotypic data with very little phenotypic data. So the Robert Wood Johnson Foundation last August gave us a nice grant to ask, how do you engage the citizens? How do you make it so that actually the public is not just as a patient, but can be engaged somehow in the research and as funders? And sort of a simple concept is, how do you make it so that data that's being generated by citizens, objective, subjective data, can be shared among themselves, but where you can move it over into very large sets of data and then allow data-intensive science approaches to be able to query that data. So one of these projects involves 3 people at the UW. I have Andrew in the middle, but Mark Schubert and Ray Monnet have been key on this. You may know this bone marrow failure syndrome, Fanconi anemia. And we thought that it would be helpful if we could see if the patients could help on the surveillance for second tumors by taking photographs of their mouth instead of going to a doctor, and having narratives that went with that objective data, and have blood counts where they tracked their blood counts and said, I'm having stress, or I haven't had sleep, or in my menstrual cycle, or I had a viral infection. We think that citizens should be in a very good position. They're much more motivated. They've got serious time, and I think they have really good insights. So we're building up an FA project. The last of these bridge projects, though, I think puts things in perspective. On your iPhone, smartphones, the microphone in them is so sensitive that we can basically monitor the variation in the gait of vocal cords the same way as a patient with Parkinson's has a variation in their gait and when they're walking. Their voice is basically one of the best ways to look at their disease. And Max Little, looking as crazy as he really is up here, at MIT Media Lab, figured out a way in a 1-minute recording to have an assessment that was 95% accuracy, 99% specificity, objective, which meant that instead of going to a doctor's office to get evaluated for your Parkinson's, now we can do real-time evaluation. We can take that objective data. We can ask the patient, what are they doing that makes them better or worse? And go to something I think is really important, which is, how do you turn anecdotes, what someone feels strongly is making them better or worse, and how do you mine that for signal? Well, you do that by having 10,000 records every day over a year, not by having a classical epidemiologic study. So I've been talking about enabling teams of teams. I talked about Synapse, Consortium, open challenges. I talked about Bridge. I did not talk about portable legal consent, because I want to end with a 5-minute description of a project that I hope will interest you, and maybe you can help us in the design. So I assume you know what these are. These are the emblems for superheroes. And what we got to wondering was whether, in fact, almost all of our research dollars are going to studying patients who are sick, trying to find defects in hopes of finding medicines. But wouldn't it be interesting if actually what we should be doing is looking at normal people and finding those extremely rare normal people who are actually walking around with otherwise, if they had the classical deleterious mutations that would have made them present when they were 2 to 5 years of age. So this project is one that assumes that we could go and take 150 or so pediatric inherited disorders, Tay-Sachs, CF, ataxia, you can go through. There are about 150 that are so severe and have such a high penetrance that you have that particular homozygous deletion, you should have presented to a hospital before you're 10 years of age. And said, let's say if you're 40 years or older, we'll take a saliva sample from you. And for less than the cost that it takes to get it to us, we can now analyze 100 megabytes of DNA, which is enough to go through all the exons for all of those genes, looking for the mutations that we know are deleterious, we know the penetrance on, and look for those rare individuals that don't show up in the way that you would expect. Because if you want to build prevention, you want to build preventive therapies, it seems to make more sense to do that. So we have sort of figured out the goal. We've figured out sort of ways to set up where things are. And we've been this pretty far. And then this is back to my beginning on talking about being naive. Never think that you have an idea that hasn't been thought of already before. And always when you're working on something, go and talk to the people who really have been thinking of it much longer than you have before you go into the design phase. So I've been lucky enough to know Maynard Olson for decades. He was on our board at Rosetta. I got a chance to check with him recently, and I hadn't found this. But there's a beautiful article from 1999 where the full concept of this, not like half concept, but the entire concept of what I just told you had been completely laid out on the power of loss of function genes and why we need to have this buffering capability. A little sidebar information, if you remember, Lee Hartwell and I worked on synthetic lethality, and there, across the whole genome, you have like a handful of genes that are synthetic lethal. When you go to what I think is worth calling super vitality, what you find when you do the same experiment is hundreds. So again, go find your giants that lie around you. Lee Hartwell, Barbara Garvik, and John Hartman did the spectacular proof of concept experiment for this superheroes project in yeast when they took 3 alterations, 2 in amino acid biosynthesis, 1 in nucleotide reductase, needed to build out nucleic acids, and crossed those defects with all the non-essential genes in yeast and found 200 around. 200 individual point mutations are able to buffer the defect as strong as crippling an amino acid biosynthesis or crippling nucleotide biosynthesis. So I think what that tells us is that genetics is going to be my whole story has been genetics is very much context specific. We have no idea what we're doing. And we've got to come up with ways to get to the rules. We have to get ways of working together to find what those rules are, because I don't think it's sort of too much of a stretch to think of a device that sits on your phone, smartphone, and actually tells you whether what you just did was taking you to a new energy minimum towards a disease or away from a disease, the way we use GPS to do physical locations. So I think to get there, we've got to look at this mismatch between the state of institutions that we have and the state of the technology. We've got to not work in little silos trying to get tenure and not thinking of ways to actually work together to do that. We've got to bring in the citizens and their insights, and we've got to change these sort of guild-like structures that have been running things for a couple hundred years. Thanks very much.